In this episode, I talked to Dr. Sarah Palmer, a leading HIV researcher and the co-director of the Center for Virus Research at the Westmead Institute for Medical Research in Sydney, Australia. Sarah's career has taken her to many countries, from Sweden to South Africa to even China and the US and now Australia. She's done all of her work on HIV, and she focuses on finding a cure for HIV. This episode here is very scientific. Sarah does a phenomenal job in breaking down all of these scientific concepts in easy to understand ways that really helped me understand how we can ultimately find a cure for HIV. We also touch on preventative measures and why it's so important to get tested for HIV. We also take a global perspective and talk about her work in South Africa and China, which is super interesting. And it also helps us understand current day stigma with HIV in many parts of the world. I learned a ton about HIV here, and I hope you do too. You are now listening to the Gustavo Franco podcast, where we challenge the norms and uncover the truths of our healthcare system. I'm your host, Gustavo Franco. Sarah, it's great having you here. In our phone conversation last week, one of the most amazing things I thought was your ability to explain really complex things in simple terms that I could understand really well. And that's why I think I was super excited to actually record this episode, because we're going to talk about some complex topics. But I'm really looking forward to see how you explain all of them. And I'm looking forward to try to understand everything and how everything works. I do want to take kind of a bigger picture look here at the beginning. I know we're going to be talking about like curing HIV. Why do we need to find a cure for HIV, and why have we not found one yet?
Okay. Well, thank you, Gustavo, for this opportunity to speak with you today about HIV, a topic that's very dear and near to my heart. I've spent over 20 years researching HIV, and it's really an important virus and infectious disease for the whole world, to be honest. So the problem with HIV is that when it infects an individual, it actually infects immune cells. So it infects CD4 T cells, which are what we call helper immune cells. So it inserts its DNA into that cell that it infects, and it takes over that cell and converts it right into a factory of producing viruses. And so first off, the virus is infecting immune cells. So those are the cells that we typically use to fight viruses, but it is infecting them and then destroying our immune cells. So that's the first thing that HIV does. But secondly, when it replicates, HIV makes a lot of errors in its replication. So what an individual is infected with, if they're not on treatment, the virus builds this huge swarm of variants. So very genetic, diverse variants within an infected individual. And that diversity can be up to 5%. And as we talked on the phone, just to give a context to that, 5% genetic diversity is the diversity between a human and a mouse. But this is what HIV does within the infected individual's body if they do not go on therapy. So that's what's made it so difficult to develop a vaccine for HIV, because A, it infects our immune cells, and those are the ones typically we enhance with a vaccine. And secondly, it's very genetically diverse. It's one of the most genetically diverse viruses in the world. So that's why the antiretroviral therapy that's been used to treat HIV infection, that's why it's been so important, because it's knocked this virus down to very low levels in an infected individual. And that's important because then that makes that person less likely to pass on the infection. In fact, it reduces that incidence to essentially zero if they are on effective antiviral therapy. However, do you have a question?
So I was going to say on that point, right? Mm-hmm. Even though with antiviral therapy, we can prevent them from first infecting other individuals, but also make sure that they live a healthy life. The reason why we still want to find a cure is that it costs a lot of money to keep people, and then also they don't want to be taking meds every single week, right? Or like an injection twice a month.
Absolutely. So for some people, taking these medications is quite difficult. It's chemotherapy. So there are some side effects from the chemotherapy. But secondly, taking a meds every day is expensive. Some countries can't afford medications for all of their infected individuals. For example, there's certain countries that only about 30% of people have access to therapy. So we really do need to cure this virus if we can. And why doesn't the antivirals, why doesn't that cure it themselves? Well, that's because they actually stop the virus from replicating. It stops different phases of the replication cycle of the virus. But there are some cells, CD4 T cells, that are infected with the virus. They go into a resting phase. So the drugs are not active against the virus that's in those resting cells. So the virus has integrated its DNA into those resting CD4 T cells, but they're just in a resting phase. And they hold the virus, and they can keep the virus for years. But antivirals have helped people to live almost a normal life. It's extended life for everyone who starts an antiviral. And if they maintain their therapies, their antiretrovirals, they can have nearly a normal life. The only issue is they do have this remnants of virus that's in these resting cells. So if they stop their therapy at any time, the virus can come right back and within 2 weeks be at a very high level in their body, and they would be infectious again. So that's why antiretroviral therapy is a lifelong therapy, because we don't have a cure at this point for HIV.
So these antivirals, is the reason that they only detect active HIV-infected cells because they detect the protein that's created by the virus, or why can they not detect the cells that are in that resting phase?
Yeah. So antivirals actually targets different stages of the replication cycle of the virus. So as long as it's replicating in a cell, the antivirals attack that and stop the replication. But for those viruses that have injected their DNA into the infected cell, but that cell's gone into a resting phase, so the virus isn't actively replicating. It's just sitting there very quietly in the DNA of that cell. And then when someone stops their therapy, and if that cell wakes up, it can start to produce virus again. But it sits in this very resting phase with containing the HIV DNA in the DNA of that infected cell. And so that's why the drugs just are not active, because there's no virus being produced in that resting cell. And those resting cells can sit in our body for years and years and years. And that's why if you stop therapy, say 5 years down the road, if I started therapy and I stopped it in 5 years, one of those resting cells could reunite a whole new infection because it has the HIV DNA in its DNA.
Yeah. And so when you mentioned that the HIV is active and replicating, does that also include like transcription to form proteins?
Okay. Exactly, it does. So when it's active and replicating, it is producing proteins. But even in some of these resting cells, there are a few proteins being produced, but not enough to really make the drugs that active against these resting cells that contain HIV.
Got it.
Yeah. So
one really interesting thing that you brought up is how the virus integrates itself into the DNA, right? HIV integrates itself in our DNA. How exactly does that work? And how do you get such variants in the actual viral DNA?
Yeah, so what HIV, it's an RNA virus, but when it infects a cell, it converts itself using its own enzymes into DNA. And then it has its own enzymes that helps it integrate its DNA into the DNA of the infected cell. But what's the problem is that the viral enzymes that come in with the virus when it infects the cell, with the RNA, when it infects the cell, those enzymes make a lot of errors. And so they're not perfect. They're viral enzymes. Our cell enzymes, when our cells replicate, we have enzymes that help to replicate DNA, and they actually have what we call a editing mechanism, so that if any errors are made when our cells replicate, those errors are cut out. HIV has its own enzymes that help it convert its RNA to DNA, but that enzyme that converts the RNA to DNA makes a lot of errors, and it has no editing mechanism. So every time that RNA is converted to DNA, there's usually one or two errors. And then that DNA is inserted into the infected cell DNA. And that's what builds that swarm of viruses, because every time the RNA is converted to DNA, there's a mistake made, typically.
Got it.
Yeah.
And usually when you get these mistakes, does that viral DNA still produce viral proteins?
That's a great question. Sometimes it can, and sometimes it can't. So sometimes that error is fatal, even for the virus, but still integrated into the DNA of the cell. So you can have a lot of dead-end virus in infected cells, but it's not going to really produce active virus. So it's those cells that contain genetically intact virus that can produce then a new infection when someone stops their therapy.
And the virus that's mutated, let's say, might not work as well, can that still replicate itself?
It can still produce some proteins. It can still take over some of the cellular mechanisms, to produce proteins, but it may not produce an infectious virus. But those proteins are still a problem because our immune system actually recognizes those proteins and becomes a bit activated. So in HIV-infected individuals, even though they're on therapy, their immune system's always just a little bit inflamed or turned on. And that's a problem because that can lead to some heart defects and heart issues or other medical issues. But so even though the therapy extends a life and you can live a long life, if you're HIV positive and you start your therapy, your immune system's always still a little turned on because there are these cells that have some dead-end virus, but they're producing some viral proteins, and your immune system recognizes that.
And that just causes some like chronic inflammation.
Chronic inflammation, yeah. And chronic inflammation's tough on our body. It's something we should all try to avoid. A lot of it can lead to heart issues, and so inflammation's not a good thing for us in the long run. Yeah.
Yeah. So you get this viral DNA that's, completely different. As you said, 5% different, which is very different, more than a human and a mouse, in all these different cells. Is that why actually finding that viral DNA is so difficult?
You can find it if you have the right assays, which we have developed an assay that allows us to sequence HIV, the full genome of HIV. So that allows us to identify those cells that contain HIV.
Would that only be the intact form of HIV?
We can find both intact and defective using this sequencing method. So we can say, okay, these cells have a lot of defective virus, but these cells have the intact virus. And that genetically intact virus is really key because we need to target that to cure patients or to cure someone who's HIV positive. So we really do need to be able to target genetically intact HIV to cure a person who's living with HIV.
Yeah. And I think this goes pretty well into the research that you're doing, right? Mm-hmm. If you had to give an easy to understand overview of exactly like what you've been doing, how would you describe that?
Sure. Well, the first thing we did was we developed a full-length sequencing method that does allow us to sequence the full genome of HIV so we can really see which sort of CD4 T cells. So CD4 T cells, the ones that HIV infects, they come in a lot of flavors and they're found all over our body. So we really wanted to determine which type of CD4 T cell contains the most genetically intact virus. And we think we have found that, which is really exciting because we know that then we must really target that type of CD4 T cell to cure someone who's living with HIV. But secondly, using our sequencing, we've been able to really, and converting that sequencing into protein modeling, we can really identify essential regions of HIV proteins that are essential for the virus to survive. And now we're actually making those proteins, and they're called peptides, so they're small regions of proteins, and we're going to use that to put into a vaccine. And what we'll do with this vaccine is that because we use these protein segments to actually educate killer T cells. And these are T cells that actually go after infected cells. They're a different type of immune cell. And we're actually using these proteins, these really essential proteins for HIV to replicate. We're using these essential protein blocks to educate killer T cells and actually help them recognize HIV-infected cells in the body. And then hopefully we can go after those few remaining infected cells and cure someone.
cool. That was a lot right there.
Yeah,
That was a lot right there. Let's break down. Let's talk about the first part first. So you mentioned that there are different types of CD4 T cells, which are the cells that are infected by the HIV. And you see that in one of these specific type of CD4 T cells, they're more likely to have the actual intact version of HIV.
Exactly. They're called effector memory CD4 T cells. And we have identified that this type of CD4 T cell contains a lot of genetically intact virus. So that's really important because that tells us, okay, we really need to target those types of cells with any sort of curative strategy.
Right. So is there a reason why these specific CD4 T cells prevent the virus from mutating as much?
The virus is still mutated in those cells, so we find intact and effective, but we find predominantly intact. And I think the reason for that is when those CD4 T cells go into that dormant phase, the effector, this is going to get really difficult and technical, but we can try. Effector memory CD4 T cells, they proliferate easily. So when they proliferate, they just copy that HIV genome that's stuck in their genome. And then they just keep proliferating. And as long as there's genetically intact virus in there, then they actually copy that along with their own proliferation. I hope that explains that.
Yeah. Does that happen in normal CD4 T cells?
So effector memory T cells are the most proliferative. Yeah. And so they are sort of the ones that really respond very fast to some sort of other infection. So they're the most proliferative cell of the CD4 T cell subsets. And I think that's really why we see so much more intact in effector memory T cells. Yeah.
But do you also see intact virus in other types of
We
CD4?
do, but at a lower level. Yeah.
Okay. So say we can target, they were called effector?
Effector memory CD4 T cells, yeah.
So effector memory CD4 T cells. If we can target these, what would that really mean? Like say we can get rid of all of them that may have this intact virus. What does that mean for the other type of CD4 T cells that still have the virus? Intact?
So you'd have to target those first, but then if there's some remaining intact virus, then go after other cells as well. But one curative strategy is to reduce proliferation just for a moment. Let your other immune cells catch up so that they can fight HIV. And so there's one strategy is to stop the proliferation of CD4 T cells. You do need those cells in your body. But if you could slow down that proliferation, you could slow down the spread of genetically intact HIV. Yeah.
And would that come in place during antiviral therapy?
Absolutely. So what happens is during antiretroviral therapy, these memory cells are in this dormant phase, but every now and then they wake up to respond to some other infection, and they proliferate. And if they have HIV in their DNA, they're copying that at the same time. And especially for effector memory cells that seem to have a lot more of intact, they're proliferating that intact HIV as well. And they may not be producing the virus because they're proliferating for another reason, but they are copying it when they copy their own DNA.
Yeah. So say we want to find these cells that have intact virus. Is it better to go after these effector memory CD4 T cells, or is it better to, because you said we have found the exact sequence of the virus, right? Or is it better to go after just that exact sequence?
Well, it's tricky because, again, you have that swarm of variants. So even within effector memory T cells, that genetically intact virus is genetically different in each one of those cells. I don't know how to explain this, but the effector memory T cells come as a bunch of cells, right? And some of them respond to this infection, some of those respond to that infection. They're memory cells. They're sort of, if you've been exposed to an infection, these are your memory cells that fight an infection if you're re-exposed. And so some of them respond to one type of infection, some respond to another type of infection. So what was your question?
I think it was mostly is it better for us to target CD4 T cells or target,
No, do they have one type of virus or do they have a different? No, go ahead.
Like, is it better to target effector memory CD4 T cells, or is it better to try to find cells that have the virus in them?
I think it would be good to do both. But if we could specifically target effector memory T cells, we could reduce a lot of that genetically intact virus, and then maybe we could have less of genetically intact virus within an infected individual, and then think of other ways to remove those last remnants of the virus. But with the work that we're doing here, we're actually developing what we call a T-cell vaccine, and that would go after any T-cell that has HIV, because we are identifying these protein regions that are absolutely essential and critical for the virus to survive. And so if we can then educate our immune system to recognize any infected cell that contains this type of the virus, this portion of the protein, and go after that, then we could possibly cure a person.
Okay.
That's down the road.
Yeah. No, but I think that's really cool right there.
Yeah.
So we now know the exact sequence of proteins that the virus really needs to work.
Yes.
So even though we might have virus that's mutated, we can still see if they have these regions for proteins that they need to work, right?
Exactly.
So now we can distinguish between virus that's going to work and virus that's not going to work.
Exactly.
Okay.
And then you can target that virus that would work and produce a new infection with a T-cell vaccine. So what we would do is we would probably make it into an mRNA vaccine, and we would then educate what we call our CD8 T cells. Those are killer cells. And we'd educate them to recognize any CD4 T cell that contains this essential protein region of HIV and attack them and just clear them out of our system.
And could that lead to complete elimination of the virus in the body?
That would be our dream. But we have a long way to go before we get there. But yeah, that would be our dream. And I've been working with very brilliant postdoctoral fellows. The person who's helped me design and identify these essential regions is Yuno Kali. And she's built what we call an immunoinformatics analysis pipeline, which has allowed us to really identify these essential protein regions. And we've identified them in 5 different HIV proteins. So we could build all that into a vaccine. Yeah.
Wait, so can you explain exactly how that works?
So you would take the peptides, which are part of proteins, and convert them into an mRNA vaccine, and then inject that. And that would stimulate CD8s because it's a foreign protein. And the CD8s would recognize that foreign protein, and then it would start to clear any cells that have that foreign protein and that foreign type of piece of HIV.
I think that the learning of the immune system, right, because that's basically what it's doing here.
We're educating it.
Yeah, the immune system's learning what cells to attack.
Yeah.
How does that work? How do they actually figure out what to do?
Oof. Well, on an infected cell, they have what we call a MHC, a major histocompatibility. Oh, that's a long word. But what infected cells, what they do is they actually put proteins of the virus on the outside of that infected cell. It sort of puts it out there. And then CD8 cells have another receptor. And if they see something foreign on a cell, they come in and attack that cell. So we're just saying to CD8 T cells, if you see a protein that looks like this on the outside of a cell, kill that cell. And so the CD8 T cell will come, bind to that cell, and destroy it. It releases certain enzymes and reagents called granule enzymes, that would kill the infected cell.
Yeah. So you mentioned that this would be likely an mRNA vaccine.
Yeah.
Why and how, what are the other types of vaccines, and why are we picking an mRNA here?
Well, I think mRNA vaccine, the major reason to pick that is because we could put as many segments of these essential proteins into the backbone of that vaccine. It can carry a very large load. So you could put all of the different essential protein regions into one backbone of an mRNA vaccine, and essentially just introduce that into the immune system all in one piece.
So then you can give it to this CD8,
Yeah.
is that what they're called? So
They're killer T cells, we call them, yeah.
So you give this sequence with
The protein.
all the possible variation of proteins that make the virus work, and then the killer cells, they're going to recognize any of those.
Yeah.
Okay.
So the killer T cells will say, oh, if that's a foreign object. And if I see any cell that has that foreign object on the outside of it, because what happens with infected cells, they do actually put some of the viral proteins on the outside, and that's how our immune system recognizes infected cells, essentially. And so the CD8s, we educate the CD8s and say, look, if you see any type of proteins that look like this, these HIV proteins, kill that cell, attack that cell. And that's what we're hoping we will do.
Yeah. And would that work for inactive cells that might not have those proteins on the cell surface?
Yeah, so that's a problem. But those dormant cells, they do produce some proteins. So that will be a problem for us. So one way we may get around that is actually to use what we call a latency reversing agent. So an agent that would wake up those dormant cells. They would produce the proteins, and we'd have the CD8s already primed to go right after those infected cells.
Okay. No, Sharon was telling me about this, I think.
Yeah.
So they're kind of like two steps to the equation, right?
Mm-hmm.
We got to activate the dormant cells.
That's right.
And then at the same time, train the immune system to be able to detect all of these cells that we're activating and then kill all of them.
Yeah.
And then hopefully in the best case scenario, HIV is gone.
Cleared. Yes.
Okay.
That's all of our dreams. So Sharon and all of us are working very closely together. In fact, we collaborate on a lot of this work. But yes, so that would be, Sharon is an expert on latency reversing agents. So we would tap into Sharon's knowledge and say, okay, possibly we could combine this vaccine, this T-cell vaccine. We call it a T-cell vaccine because it's targeting the CD8 T-cells and helping them to recognize these proteins. So we would use this T-cell vaccine in collaboration or in concert with a latency reversing agent. Yeah.
So where are we right now with latency reversal agents and with this mRNA vaccine?
Yeah, so latency reversing agents, there's been several that have shown to actually reduce latency and that you do see a virus come out of cells. So there are some that are quite promising, but I think we have a ways to go with that because they're not waking up all of the dormant cells that have HIV. So we do need to work on that side of it. But as far as identifying the essential reasons, we've identified those. We are now testing to see how well they do stimulate CD8 T cells. And we're doing this all what we call ex vivo. So we have samples from people living with HIV who've been very generous and donated samples to us. And we are stimulating their CD8s with our protein constructs and our essential protein regions to see do they actually go after infected cells. And so far, it looks quite promising.
So, that's cool. So is this basically a form of the vaccine already?
Yeah, we're looking at different segments. So we're looking at different protein regions right now. We haven't put them all together in one group. So we're looking at we have 5 different proteins and the essential regions from those proteins. And we're testing all 5 of them right now. Eventually, we want to put them in a big pool and see how that goes too. And this is all work that's actually funded by the U.S. National Institutes of Health. They've given us a couple of grants because they're quite interested in to see, does this work?
Yeah. Yeah.
So, yeah.
So let's say we are able to put all these proteins together and create this mRNA vaccine that works really well. Mm-hmm. And we're also able to find a latency reversal agent that works really well. Mm-hmm. After that, what are some problems that we still might run into in completely eliminating the virus?
Sure. I mean, maybe the latency reversal agents don't wake up all of the dormant cells, so there could be just a few cells left. It only takes one infected cell to reinitiate a whole new infection in someone living with HIV. So the way to test that, to be honest, would be to stop all therapy to see if the virus comes back.
Okay. That's tough.
It is, and it's a lot to ask of a person living with HIV. It is. But we have a lot of generous people living with HIV who are willing to go through some of these clinical trials where they stop their therapy, and that allows us to see how effective any curative strategy is.
Yeah. And would the latency reversal work for cells that also have defective versions of the virus?
Yeah, they could produce some proteins, but you may still have some cells that have defective virus that we never really eliminate. But because it's defective, it wouldn't cause a new infection. The only thing it would cause is some inflammation.
Yeah. But could that defective virus mutate back?
No.
Okay.
No, there's no way that it can do that. So once it's that defective, and typically when HIV, when it replicates and it produces defective virus, they have huge deletions in their DNA. And there would be no way that they could ever recombine or anything to become infectious again.
Yeah. So, I mean, it seems like we're at a pretty good spot with after all the cells are activated, detecting these cells and being able to kill those cells. So when it comes to the latency reversal, why are there so many difficulties in being able to reverse all those cells?
Yeah. So a lot of the latency reversal agents come from cancer therapies. And we know the concentration of those drugs that are effective for cancer treatment. We don't know what the concentration is needed for HIV, reversing HIV. So, and a lot of the times we use very low concentrations of these latency reversal agents to be on the safe side. So maybe we have to increase the dosages, but it would have to be done very, very, very carefully. Because again, we're treating people living with HIV who are already on antiretrovirals. So how would those drugs interact? We have to be very careful about all of that.
And do these agents just tell the cells to replicate the DNA?
So when a cell is resting, its DNA and its chromatin is very tightly bound. And what latency reversing, they just open up the DNA and allows the cell to replicate again. But when it's in a dormant phase, the DNA is kind of in this very tight bound formation, allows that to open up.
So the goal would be to find an agent that will get all of that DNA that's
tightly bound,
and then open all of it up, and then we can use this mRNA vaccine, come in and kill all of them.
Yes.
And there we go.
But I must say we are a long way from there. Yeah. But we're all trying different avenues because all of us know the only way that we can cure a person living with HIV is remove every remnant or every cell that contains replication component virus, and just remove all of it replication component virus. It only takes one cell with one replication component virus to reinitiate a whole new infection.
Yeah. So Sharon mentioned that there have been a couple of patients that were able to completely eliminate the virus, right?
Yes.
So how did that work?
Yeah, first off, that's something we could never do globally. So there's some people who have a genetic modification on their cells, which makes their cells less susceptible to HIV infection. So their cells cannot be infected. There was a bone marrow transplantation. They took cells from a person. There's only about 0.1% of people in the globe that have this modification on their cells that makes them absolutely resistant to HIV infection because the virus can't attach to their cell and infect them. So they took cells from someone who is absolutely resistant to HIV infection, and they did a bone marrow transplant into a person who had cancer, but was a person living with HIV and had cancer. And they transplanted those cells that are resistant to HIV infection into this person's bone marrow. And when their bone marrow with these cells, they were resistant to HIV infection.
Okay, interesting.
Yeah, it's actually amazing. But one thing about bone marrow transplantation, 20% of people pass away when they go through a bone marrow transplantation. It's only done in very, very sort of desperate cases where this was a person living with HIV who had cancer and needed a bone marrow transplant. And it cured that person of there. And that was the Berlin patient, the original one. The clinician for this person living with HIV or the Berlin patient who had cancer said, I'm going to look for a bone marrow match from someone who has these cells that are resistant to HIV infection. So really did this on purpose, and it worked.
That's quite amazing.
And for us in the field, we're like, there is a way to cure. It really gave sort of a bolus into the field that we can cure this disease because we've all been realizing that that's what we need. As great and as life-changing as antiretrovirals are and how they've allowed people to live a very long life, access is a problem. So only about 70% of people in the world have access. And sometimes in some places, it's a lower percentage of people who have access to antiretrovirals.
I think it would be interesting to kind of take a global look here. I know when we were talking, you said that you did a little bit of work in, I think it was Kenya, South Africa, and China in relation to HIV. So how are those countries dealing with it?
Yeah, China's doing quite well now. But when I was working in China, it was in 2003. And at that time, the Chinese media called HIV a foreigner's disease. And they really did not believe that they would have any issues with high infection rates of HIV. But then the virus, and it's a virus, as we know, did initiate an infection in China. there was a blood transfusion infection that happened in multiple towns. And so then the infection just started to spread in China. And there's also a little bit of injecting drug use in China. So a lot of injecting drug users were also exposed to the virus. And so at first, the Ministry of Health was quite worried, but they were having a hard time convincing other ministries in the Chinese government that we are going to have a problem with this virus. So we went into China with a consortium to try to talk to the Chinese officials and other ministries, not only in the Ministry of Health, they knew they had a problem, but the Ministry of Education, the Ministry of Policing, all different ministries. We really to help educate them about this virus, and it's not something that you can stop at your border, and it's not a foreigner's disease. It's actually a disease that's spreading in your country right now. And so that was really important. But what helped us, sadly, was the first SARS epidemic in China, because I think the government was not that worried about HIV because it wasn't economically hurting their country. But then the first SARS-CoV-1 infection happened, and that really hurt the economics of China because nobody wanted to go there, nobody wanted to do business. And then they realized, you know what, a virus can hurt us as a country. So then they really started rolling out treatment for HIV.
So why was China specifically reluctant to recognize HIV as a national threat?
I think they just didn't want to believe it. I just think they thought, no, we're not going to have this problem here. Or if we do have this problem, it will be in marginalized communities, sex workers, injecting drug users, foreigners. So they really didn't want to believe that they would have an issue with this virus. Also, I think it's the mode of transmission. It's a difficult discussion to have. HIV is typically sexually transmitted, and so it's a mode of transmission for this virus that a lot of people don't want to have a conversation about.
Yeah. And do you think that the government just didn't want to believe they had this population of sex workers, or they had these communities that are more likely to actually get the virus?
Yes. And I think also there are certain groups, certain towns that are known for their homosexuality in China, and that was also not accepted. That was taboo. There's still a lot of stigma associated with HIV in a lot of countries. So China definitely had a lot of stigma attached. And that was another reason we went with this consortium. We tried to say, you know what, we're all struggling with HIV. So let's just take away the stigma and let's treat people who are infected. Yeah. And I guess similarly, that was the same for South Africa. Some years ago in South Africa, the leadership did not want to believe that HIV was a problem there as well. And they wouldn't even allow treatment to prevent mother-to-child transmission. If a mother is treated with antivirals and she's pregnant, then she will not transfer the virus to her unborn child. And in South Africa, they wouldn't even allow that. This was some years ago. South Africa has absolutely changed. Every country is different how they approached HIV. Some countries approached it very well. Botswana was one that really the health ministry started treating people right away, and they have had less of an issue than other countries within the continent of Africa.
In terms of resources, do they have the same access to resources that a lot of, like, say, the US and Australia have?
Not always. There can be some delivery stoppages. So, for example, and these drugs are expensive. So for a lot of countries, it's really difficult to provide lifelong antiretrovirals. We're really lucky here in Australia. The government provides lifelong antiretrovirals to any HIV-positive person. They have access to that. But not all countries have that. And that's why we need a cure.
Another issue that I just thought of with some of those countries is that they really don't even recognize homosexual communities, right?
Exactly.
And I mean, I think that's still a problem to this day.
It is.
So how does that really play a role in HIV treatment there?
It's very difficult because these communities, first off, are very worried to be identified. And so they do not want to go to clinics if they possibly think they are positive. So it's really hard. Stigma is that's what has hurt us a lot with this virus, is stigma. There's stigma attached to it. And we have these great treatments now that we could help people and prevent new infections within a community. And people are afraid to go get tested. So I think the most important thing for someone to do is to know their status, their HIV status. Because once you know, then you can start taking therapies if you have access to them. And then you would prevent spreading the virus to other people within your community and loved ones. Yeah.
Yeah, because I think it's so much easier for us to recognize that there was the stigma in the like 80s and 90s in the US, but kind of because we live in those communities now, we're like, oh, the stigma doesn't exist anymore. But in many parts of the world, it's still there. Yeah.
I worked with a clinician from South Korea, and he said, it's so funny. And at that time, I was working in the States. And he said to me, it's so funny that there's so many homosexuals in the United States. We don't have any in South Korea. And I was like, okay. I didn't know what to say at that point. But I was like, well, I think you may have some communities, and they should be as educated as any other community.
Yeah. So is the reason that it's important to recognize these communities because we would likely want to have like more screening for them because
they're more at risk, but also more education. And then also there's other treatments like what we call PrEP, pre-exposure prophylaxis. So they could take some antiretrovirals if they thought possibly they may be exposed. We also have post-exposure prophylaxis, so that if someone's had an encounter or a sexual event that possibly they were exposed to HIV, then they could take treatment right away. And that has prevented a lot of infection. So if we could educate everyone that, hey, there are options. You don't have to take antivirals all your life. You could take pre-exposure or post-exposure.
Yeah. So I think prevention is a really cool topic. Sharon and I kind of touched on it, but I do want to talk a little bit more about it. So the reason that screening is so important is because even though you may have the virus, you might not develop AIDS until like 10 years.
Exactly.
So you could be infected with the virus, not know about it, and be spreading it to other people. So that's why we kind of like should be screening a lot of people to detect that virus and put them on antiviral therapy so that they can't spread the disease more, right?
That is absolutely right, Gustavo, because if we can identify everyone that's HIV positive and put them on retroviral therapy, we could possibly break the chain of infection. That's something that they're attempting to do here in Australia, but I mean, it's very difficult to do. But you're absolutely right. With HIV, if you're living in a developed country, developing countries are a little different, but in a developed country, you can live with that virus for up to 10 years and not even know it. And then your immune system starts to fail because it does attack your immune system. But you can live for 10 years and not even know that you're HIV positive. So knowing your status is really, really key. And there's no shame in that. I mean, I think the more we're tested, the safer we all will be.
Could technically, like in the perfect case scenario, if we can screen everyone often, just like completely eliminate the virus because we're stopping the spread of it?
Yes, we could bring it down to zero.
Do you think that that's harder to do than finding a cure for it?
I think it would be very hard to do, yeah. There would still be people who are very, very worried about the stigma so it will be very difficult to do. You'd have to have a very open society and a very open country to be able to do that. My dream is we wish we could do that,
Yeah.
But I don't think that's feasible, unfortunately. I know in Australia, that's a huge campaign. Can we get to zero? And that's exciting. It would be great if we could, but I'm not sure that will be the case.
Yeah. So in terms of screening, should, say, everyone be getting screened if they're sexually active like every year? Or are there specific groups that we want to target to actually get screened? How does that work?
Yeah, I think that's a very personal question up to the person. But if you ever feel like you've had an exposure in any way, whether it's through injecting drugs, through sexual activity, you should get yourself screened. So I wouldn't say just do it every 6 months. I would just do it whenever you feel like possibly you were exposed. If you had sex without protection and you didn't know that partner, then that might be a good time to get screened.
Yeah. And I understand that the accessibility to testing is super important here,
Yes,
right?
It is.
And also just feeling secure to go somewhere and get tested without other people having to know. Because I feel like a lot of people would want to do it in secrecy without other people knowing, right?
Sure.
So has that become a lot more accessible recently?
It has actually. So there are tests that people can do at home. I think that's great because that gives them that security of maybe I can just do this privately. And I think we need more of that availability. And I know that in China, for example, for HIV clinics, they would try to hide the name because they didn't want people to know that this was an HIV clinic. So that's hard, isn't it?
Yeah. Something else that you mentioned that I find really cool is and PEP. I had no idea about this before talking to Sharon. She mentioned it briefly. If you take this before, you can have a sexual encounter with someone that's HIV positive and not get the virus. Or if you have a sexual encounter with someone that is HIV positive, you can then take PEP and not get the virus.
Exactly.
So how do they work? And who should actually be taking them? Because I bet a lot of people wouldn't know that they had an encounter with someone that could have the virus.
So what this is just several different antiretrovirals in a combination. And you just take it. And then if you think you're going to have a sexual encounter with someone possibly HIV positive or someone you don't know, maybe after a party, whatever. Yeah, you would just take the PrEP and you would take it at a certain time before you have that exposure. And secondly, with post-exposure, you should take that immediately as quickly as possible. So you should have it in your medicine cabinet.
Because that prevents the virus from replicating.
Yes, it does. Both of them do, because they're both antiretrovirals. They stop the viral replication cycle. And so if you can stop it right away before it can go into other cells, it prevents.
How accessible is this?
Here, it's very accessible. I think it depends on each country and each,
But is it something that you can just go to the chemist, the pharmacy and get?
You can.
Oh, okay.
Yeah. And typically, well, you would have, I think you may have to go to your GP first and just discuss a little bit with them, and then they can say, yep, here you go.
Are there side effects to it, or like should anyone, before they have a sexual encounter with someone that they don't really know well, be taking it?
I would say if you're young, there probably aren't many side effects. I haven't seen anything in any of the literature about serious side effects from PrEP or post-exposure prophylaxis. But I think it's a good thing for a young person to have if they ever feel like they would get exposed.
Right.
It's easier to prevent the virus than if you have it and then you have to take treatment every day of your life.
I think something else that's really interesting here is how different viruses interact together. I know Tony and I last week talked about herpes and HIV and how 30% of HIV cases actually come from people that have herpes because it makes you more susceptible to getting the virus. I know there's a similar case for HIV and hepatitis B that Sharon works with, but you also mentioned that there's a relationship between HIV and COVID. I would love to hear more about, because I did talk to Tony about.
Yeah, so with COVID, it's not so much that it increases your infection frequency, but with COVID, what happened because of the worldwide sort of shutdown, antiretrovirals were not delivered to countries that needed them. And so that's what we're worried about, is that there could be another spike in HIV infections because people did not have access to their antiretrovirals. So that was a problem. It reduced the ability for people to take their medications that they needed.
So during COVID, because we were having to push out all these COVID vaccines, the distribution of HIV treatment slowed down?
No, not so much about the vaccine development. It was more about that, like, for example, in Australia, we shut down. So we had fewer flights coming in. And so certain countries, the people weren't traveling there. Planes were not flying there. So that broke the chain of delivering of antiretrovirals. So there were some countries that their supply just diminished. So that was a problem. Yeah.
Are there any estimates as to how many people actually had to stop their treatment because of it?
I haven't seen any estimates yet, no. But I do know that it was an issue.
Yeah. And so now we're worried because many of those people that may have stopped their treatment might now get a bunch of the side effects of AIDS.
Yeah. Well, they could, if they stop their therapy and they haven't been able to reinitiate it, absolutely, they can become sick. But secondly, they're infectious again.
Oh, right.
Yeah. And that's the major issue. And especially if, let's just say, a woman gets pregnant and she's HIV positive and she has no access to her medications, then she could pass that on to her unborn child.
Yeah. And do you know anything about HIV and hepatitis B? Because I know that's something that Sharon worked with. But
yeah, Sharon and I actually collaborated on that, and we actually published something together about HIV and HBV. Well, as part of that study, we just looked to see BV infection increase the reservoir of virus in cells, and so does increase the more genetically intact virus. We didn't see that it did per se, but it did cause increased inflammation.
Okay.
Yeah.
Interesting.
Yeah. And so we collaborated on that.
That's fun. Okay, cool. Yeah. Cool. I do want to talk a little bit about the government funding. I know that you said that the NIH funds a bit of the research here. Where is most of the HIV research happening in the world? And is it usually funded by the government? And which governments are really doing this funding?
Yeah, I would say the U.S. government is extremely active, especially in funding curative strategies. Sharon and I are both part of this very large consortium called DARE, it's funded. DARE to Find a Cure is the name of our consortium. And it's funded by the U.S. National Institute of Health at $30 million. But not just to Sharon and I, but to a large consortium of researchers. And it's led by Steve Deeks from the University of California, San Francisco, and Sharon and Louis Picker from Oregon State Health University. And then they bring on board other researchers, such as myself, to do certain aspects of the sequencing, analyzing the reservoir. So it's amazing how much money the U.S. National Institutes of Health have put into HIV curative strategies, but also just understanding HIV. And also that these consortiums are worldwide. It's not just in the United States. I mean, they're funding me and Sharon in Australia. I know they're funding people in Canada. I mean, it's phenomenal. And it's really bringing together a lot of different expertise together to tackle this virus, this infectious disease. But I think it also is bringing us closer to curative strategies because we all have different ideas and we're all working together and supporting each other. And I think that's really amazing. The Australian government has also put in quite a bit of money. Sharon and I were also part of a consortium here in Australia where the Australian gave us 16 million Australian dollars. And again, it was a large consortium, but in Australia, which really helps. So any sort of funding and large amounts of funding helps combat any infectious disease, not only HIV, but other infectious diseases as well, and other diseases as well. But I would say the U.S. has been probably the most generous and the most active in finding an HIV cure or understanding HIV further.
Right. That's cool.
Yeah.
Another topic that I find interesting is like the role of the pharmaceutical companies here.
Yeah.
Well, like with HIV in general. So have there been any interesting developments with like pharmaceutical companies and HIV? What's the relationship? Is there anything that we're worried about?
I would say a lot of pharmaceutical companies, Merck, Gilead, again, most of them headquartered in the United States, really pushed the envelope for developing the antiretrovirals that we use today. If it wasn't for their funding into research and development of these antiretrovirals, we wouldn't have them. And they would also collaborate with us to test their antiretrovirals, determine whether resistance can be elicited through the treatment of the virus, if the virus can become resistant to these antiretrovirals. So in the early days when we had one or two antiretroviral, the virus did become resistant. But now that there's a whole combination of them, the virus doesn't have that capability. And that's really thanks to a lot of these pharmaceutical companies who really put a lot of money into this. I know pharmaceutical companies get a bad rap at times, and maybe sometimes maybe they should. But I do know that in the field of HIV research, a lot of pharmaceutical companies put a lot of money into developing these antiretrovirals. So we're quite thankful for that.
No, that's great. And do you see a lot of variation by country in the price of treatment?
Yes. I would say the treatment's probably the most expensive in the United States. And then other countries are now because a lot of these antiretrovirals have gone off of their, you can make the generic formations of them now. So India is making quite a bit of generic antiretrovirals, and that's actually helped their country quite a bit because it's not as expensive. Once they go off patent, then they can be made generically, and that does reduce the cost of these drugs. And they can be made locally too. So local manufacturing is much cheaper than in a developed country.
No, that's interesting. So India, U.S., are there any other countries that produce like significant amount of antiviral therapies?
I would guess China does, but to be honest, I don't know. Yeah, I don't know. But I know that India definitely makes generic antiretrovirals. I think Brazil does too, to be honest. Yeah.
Cool. Okay.
Yeah.
Yeah. So, but does the U.S. kind of distribute globally?
It was at first, but now that a lot of these have gone off their patent, they are being made. Like I said, and a lot of them are being made in India and then redistributed at a cheaper price.
Yeah. I think one of the last topics here that I find really exciting is like the role of AI in healthcare in general. I know that specifically in research, I mean, there has been a ton of it already. Specifically in HIV research, where do you see AI playing a role?
It's already helped us. So when we developed this immunoinformatics analysis pipeline, we actually relied a bit on AI to help us look at the proteins of the virus. I don't know how to explain this, but it also helped us to stack up all the sequences, and we could really look across thousands and millions of sequences to see where does the virus not change. And then we could say, okay, I'll take a protein, for example, a viral protein. Let's say GAG. GAG is a viral protein. And in GAG, we could stack this HIV GAG protein, millions of sequences, and we could say, okay, these regions aren't changing. So then we went to the proteins to see, are these regions essential for that protein? So AI does allow us to do a lot of bioinformatics that were more difficult to do. So I think it'll become more and more important as we go forward, especially as we sequence more and more of the virus, because more and more labs have the capability to do full-length sequencing. First, there's just a few of us, but now it's great. I think it's the way it should be, that once you develop an assay, you should share that assay with other labs all over the world. I strongly believe that. And so the more sequencing we do of HIV, the more AI can help us look at this virus to understand parts are so essential for it.
Right.
And then we could target those.
Because now that you can sequence the virus nearly everywhere, you can put all of these sequences together and AI can look at what the similarities, differences are.
Exactly. And we know that, across the globe, there's so many different subtypes of HIV. So you heard of variants for SARS-CoV-2, right? HIV has lots of subtypes, and different subtypes are found in more of a concentration in one area than another. So, for example, the subtype that's predominantly found in United States and Australia is subtype B. But the subtype that's predominantly found in Africa is subtype C. And genetically, they're very, very different. So AI will allow us to stack all those up and see, can we find regions where it's identical?
Okay, that's interesting. Do you think it will help a lot in, say, the reversal agents, in developing reversal agents or finding reversal agents?
Latency reversing agents?
The latency reversals.
I think, how could we apply AI for that? I'm not exactly sure. But I know that it will absolutely allow us to identify regions of the virus that just don't change. And that's what you should target with any sort of vaccine, to be honest. And that's another thing. There's the Los Alamos database. I don't know if you know of this. There's a lab in New Mexico, in Los Alamos, where they actually store all the sequences that we do of HIV. And so you can go, and it's all open access. And again, it's the U.S. government provides this. And so we can go in and access all of these sequences and look at them. And AI is allowing us to do a lot, to really do a lot of understanding of what does this virus really look like and how can we target it.
That's really cool. Something else that Sharon mentioned that we didn't go into detail was like epidemiology and trying to find groups that are at most risk of infection. Do you think we can use, say, AI and computer science to really help zone in into these groups?
Yeah, I think we could, because we could identify those that are most risk of being infected, and then we could go in and educate. Because one thing about HIV, education helps a lot. If we can educate people and say, okay, there's PrEP, there's PAP, there's, protect yourself, know your status. There's so many things that we could do to reduce a new infection.
So finding these communities is key, so then we can allocate the funding in a better way.
Yeah, allocate the funding and also educate and go in and really provide the best education we can to help them understand, how do I prevent myself from getting this virus?
Right.
Yeah.
Right. That's cool. I guess my last question here is, what would you say in the next 5 years is the number one priority in HIV globally, right? Would it be to improve treatment so that it's at a similar level in all countries? Would it be reducing stigma in many countries? Would it be making a push towards a cure?
Do I have to pick one? I think I would pick all of those. I think for me, if we could reduce the stigma and people would feel safe to come forward, then that would lead us to preventing infections, and also possibly we could discuss curative strategies with everyone. I think globally, for me, if we develop a cure, it has to be a global cure. It can't just be something that works in the United States or Australia or Europe. It has to be a cure for everywhere. So I think the number one priority for me is that we reduce stigma and then we make curative strategies global.
Yeah. No, that's great. That sounds great. Are all countries right now, do they all treat HIV, or do some of them don't even accept its existence?
Yeah. So I think, again, that comes to stigma, doesn't it? Or I think for HIV, this virus, because it's the mode of transmission, people are sensitive about. Yeah. I know there are countries that don't really talk about it, and I don't want to mention any, but yeah, because, it's the mode of transmission. With SARS-CoV-2, we breathed in that virus and we all got infected or not. But with HIV, it's the mode of transmission. It's sexually transmitted or through injecting drug use.
Yeah. I think one thing that I didn't ask is why are these communities more prone to actually getting infected by the virus?
I think it's, again, education. Yeah. And that they are already sort of ostracized as it is. So if you're injecting drug users, say in Western China, you're thrown in jail and you're not educated about maybe how you could stop being an injecting drug user. So you hide that. You hide that and you don't tell people that's what you're doing. And that's how these people are more at risk, because they hide the activity that could possibly cause them to become infected.
And why are gay men more likely to get infected by HIV?
It's the tissue. It's the mode of sexual action. So anal sex is a lot more damaging. So there's more tears in the tissue, and that's why.
Okay, that's interesting. So that puts their cells at more risk of getting infected.
Yeah. Similar to what Tony was saying with herpes virus, that causes abrasions in our tissue.
Well, that's interesting, though.
Yeah. But no, I don't have any issue with it. Unfortunately, the virus takes advantage of it.
Yeah. Cool. Well, Sarah, this was great. Thank you a lot for your time. I think you explained everything in a very comprehensive way, an expert in any of this. So this was really cool to learn about.
Well, thank you, Gustavo, for giving me this opportunity. And thank you for making podcasts. So hopefully more people will learn from us.
I hope so. I hope so. Yeah. Thank you so much. Bye-bye. Thank you for tuning in to this episode of the Gustavo Franco podcast. We hope our exploration of the world of healthcare has been insightful and thought-provoking for you. The opinions expressed here are ours and of our guests, and should not be considered professional advice. This podcast is a collaborative learning experience for both the producers and listeners. While we strive to convey accurate and up-to-date information, we acknowledge that our research may have limitations. We encourage all of our listeners to supplement their knowledge by conducting their own research. If you enjoyed this episode and want to stay up to date on our future releases, make sure to subscribe or follow us on your preferred podcast platform and social media. We highly value your feedback, so please don't hesitate to reach out with your thoughts, questions, or suggestions for future topics. Together, we can foster a more educated America and contribute to a more effective healthcare future. Thank you for joining us on this healthcare exploration.